We move on to 8 brief lectures of students. And the first one will be Gidi Karmon from Tel Aviv University. Will talk about the lead ADNP gene and ADNP syndrome mouse models as prime examples of gender differences in autism. Gidi, yours.
Hello, everyone. So my name is Gidi Karmon, and I'm an MD PhD student under the supervision of Professor Elena Gozes in Tel Aviv University. And I will talk about ADNP and gender differences in ADNP and what we can extrapolate, hopefully, to ASD. So just a brief introduction into ADNP. ADNP stands for activity-dependent neuroprotective protein, and it was discovered by Professor Gozes and the Gozes laboratory as a protein protecting against excitotoxicity and having potent neuroprotective properties. It was also discovered that when complete knockout of ADNP disrupts brain formation, and knockout mice cannot close their neural tubes, as can be seen here, and they die in utero. Further research showed ADNP is essential for brain function as a master gene regulator, regulating hundreds of genes, playing a crucial role in many cellular processes, including autophagy, dendritic spine plasticity, axonal transport, alternative RNA splicing, Wnt signaling, and chromatin regulation. And in 2014, it was discovered that mutations in ADNP are present in 0.17% of ASD cases, making it one of the most common causes of monogenic autism. These patients have distinct dysmorphisms, and the majority of these patients with ADNP syndrome have ASD, intellectual disability, speech difficulties, abnormal gait, and GI and cardiac defects. And as you can see here in a recent paper, it is further established ADNP is a major monogenic cause of ASD. Here you can see ADNP alongside with the CHD8 and SYNGAP and CSN2A and SHANK3. So now we dive into ADNP sexual dimorphisms. So ADNP is a VIP responsive gene, and VIP exhibits dimorphous sexual expression in the hypothalamus. So we investigated whether the same can be seen in ADNP, and indeed ADNP is extremely sexually dependent, as you can see here, with male expression much higher than females. And female ADNP expression in the hypothalamus changes according to the estrous cycle. Also, from data extracted from the GTX database, you can see that ADNP levels in the ovaries and the testes rival ADNP brain expression, and that there is a very high expression in other sexual organs such as the vagina, cervix, and prostate. Furthermore, in a recent paper that we published, where you can see here the graphical abstract, we found a very high correlation between ADNP and the GnRH receptor in mesenchymal stem cells, further tying ADNP to sexual maturation. In the same article regarding ADNP and muscle dystrophies, we also found, functionally speaking, that when we knock out ADNP in gastrocnemius muscles using Cas9, it resulted in a sexually dependent gait deficits, further expanding these sex differences. So, since the major part of ADNP syndrome relates to speech, we examined a highly relevant model for speech, and that is songbirds. So on the left, you can see the zebrafinch, and on the right, you can see the canary and the relative data. And only males of both species are able to sing, and not the females. And indeed, we observed increased in ADNP expression in males and not in females in the cerebrum, the part that relates to singing ability, which may indicate ADNP's role in this singing ability. And these differences between males and females was not observed in other brain areas not related to singing. And as you can see in the canary, these sexual differences were not visible in VIP. So moving on to some molecular dysmorphisms used in ADNP, we utilized our model, the ADNP plus minus mouse model, or ADNP haploinsufficient mouse models, that exhibits extensive sexual differences. First, we can see, similar to the hypothalamus, we can see that wild-type male mice, or ADNP plus male mice, show higher ADNP brain expression, and that when we have haploinsufficiency of ADNP, only males show the appropriate reduction in ADNP expression and not in females. Furthermore, we saw a male-dependent increase in eukaryotic initiation factor 4E, or EIF4E, in males and not in females, which may correlate to previous studies where overexpression of this gene led to ASD phenotypes in mice. We can also see sexual differences in the ApoE gene, a major risk factor for Alzheimer's disease, which has also been shown to be regulated by ADNP during embryonic development. And we can see polar opposites between the sexes, between the males and females, and wild-type mice and plus-minus mouse. Also, while examining splenic gene expressions, we saw an increase in KDM5D, which is a Y chromosome-specific chromatin regulator, and an appropriate reduction or amelioration when treating with our drug candidate, NAP. We've also demonstrated sexual dependencies in axonal transport measured using manganese-enhanced MRI, where the transport rate was significantly slower in males than in females and in ADNP plus minus mice compared to wild types. And we have also demonstrated sex-dependent change in Beclin-1 only in female ADNP plus minus mice, where Beclin-1 is a key autophagy gene also implicated in ASD. Another study that we performed relates to the microbiome, with microbiome being increasingly implicated in the pathogenesis of ASD. And we also detected sex-specific gut dysbiosis in the ADNP plus minus mouse. And more importantly, these changes were correlated with sex-specific behavioral alterations in the open field behavioral test. And as you can see, males and females, ADNP plus minus males and females, had distinct microbiota signature. And to expand on these behavioral dimorphisms, in the ADNP plus minus mouse model, we've also done extensive phenotyping of this model. And as you can see here, male ADNP plus minus mice have a more robust motor phenotype, as can be seen in the hanging wire test and the grip strength test. And females have a more robust phenotype in the social recognition test and odor discrimination test. Again, showing these sex differences, and both sexes show phenotypes in the object recognition test and the social memory test. We've also examined the effect of stress on the ADNP plus minus mouse, with ASD patients being more susceptible to stressogenic situations, and we induce stress using 24-hour lighting in their vivarium. And as you can see, male and female ADNP plus minus mice react differently to stress with males being more susceptible to stress in the novel object recognition and social recognition tests, and females more susceptible in the open field and elevated plus maze. So, as I hope that I conveyed in my lecture, it is well established that ADNP is highly sex dependent, and it is evident in the molecular and cellular and behavioral levels. And ADNP, I'm reminding you that ADNP is a very common monogenic cause of ASD, and ASD again being sexually dependent. And ADNP may serve a vital role for further research in this subject. And I don't know if I have a couple more minutes or no. Okay.
No.
So thank you very much, and thank you, Professor Schiffman and Professor Wagner and the organizing committee, and my PhD supervisor, Professor Ilana Gozes. Thank you very much.
Thank you very much, Gidi, for a very nice talk.